The last cytoskeleton element that we are going to talk about is actin. And unlike the other two, actin is simply found anywhere in the cell. So wherever you look at the cell, you're going to see actin, unlike microtubule or intermediate filaments. But they are more concentrated just beneath the membrane. So if you just check below the plasma membrane, you're going to see a high concentration of actin. Actin is the thinnest of all. Okay? It is so tiny compared to microtubules or intermediate filaments. Although it is so small, it is very useful because you can organize them into shapes. Okay. So we have seen that there are proteins binding the microtubules to each other, and there are proteins binding microtubule to intermediate filament like plectin, but these are some limited examples. But when we come to actin, you can make so many things out of them. You can make 2D shapes, or you can do 3D gels out of them. So the actin binding proteins are quite large and diverse. So there are so many proteins that binds actin to give the tiny shapes. So the nice thing about actin is also because they are small and they are easily manipulated, the small shapes on the cell surface, like this phagocytosis arms, they are established on the basis of actin. Think about a neuron. There's the soma and the dendrites and axon terminals. Okay. These long fragments, of course, it is full of microtubules because the transportation is needed, and it is also full of intermediate filaments because it needs mechanical resistance. But the tiny structures here in the postsynaptic site and presynaptic site is established by actin, because if you check in detail in the structures, you're going to see a very nice shape of the presynaptic site and a very nice mushroom shape of the postsynaptic site. And all these structures, they are established with the help of actin cytoskeleton. These are actin-based structures. You would not find any microtubule or neurofilament here. And any deviation in actin structure or actin binding proteins, they would also disrupt these synaptic structure, and they would cause mental retardation just because the synapse shape is not correct. Okay. There are 4 basic classes of structures made by actin in the cell. Microvillus. Please do not confuse them with the cilia. Cilia has a different structure. They are thicker and microtubule based. These are the microvilli. They are thinner and actin based. Okay. Microvilli. And these are the contractile bundles. We call them contractile because they can contract. Okay. They are like muscles of the cell. Okay. If we think the cell like a human, the bones are made of microtubules. These are the strong ones. They're making the bones of the cell. They are keeping everything in its place. And the muscles are made of actin because they can contract, so they can make the cell change the shape between these two. It can contract and release, contract and release. They can make some protrusions. These protrusions can be filopodia or lamellopodia. These are like small fingers of the cell. It is searching around what is going on around, how it is going to grow, and which direction it will grow with these little fingers. And finally, there is the contractile ring. It will take place in the M phase after the mitosis. Remember, there is a stage called cytokinesis. The division of the animal cell into 2. There are the ER and Golgi and microtubules in both of them, and mitochondria. How they divide, it is simply cut into 2 with the help of a contractile ring made of actin. Okay, let's check the molecular structure. Actin is also a globular protein, but this time it is not dimer. Remember, for the microtubule, it was a dimeric protein made of alpha and beta subunit. And think about the intermediate filament. It's actually made of tetramer. The smallest subunit is this tetramer. Okay. In case of actin, it is much smaller, much thinner. It is made of this globular protein. So it is not long and thin. It is sphere shaped, but it's not exactly sphere. There is a difference between these two sides. This is why it is also polar. Polarity is important because polarity is the basis of movement, and actin cytoskeleton, surprisingly, is also used for movement, and there is a minus and a plus end. Okay. Again, this minus and plus has nothing to do with electrical charge. It's only about growth. So it is going to grow this way. Addition of monomers, the polymerization will take place mostly here, and the depolymerization will take place here. Okay. In the actin cytoskeleton, in a cell, in a eukaryotic cell, what you're going to see is that they are mostly bound and stabilized by the actin binding proteins. If they are not bound to actin binding proteins, they will do a movement called treadmilling. Okay, it is like dynamic instability, but a little bit different from treadmilling, because remember, in the case of microtubules, this minus end is attached on the microtubule organizing center. Here, the minus end is free. Okay. When we talk about the actin cytoskeleton, they are not originating from one single place in the cell. There is no actin organizing center. Okay. It can start anywhere in the cell, or the minus end can even be free. Okay. In this case, the monomers, they will be released, they will depolymerize from the minus end, and they will polymerize here to the plus end. So the filament, it will start like this because it dissociates here and it polymerizes here. In 10 minutes you would see this, in 10 minutes you would see this, in 10 minutes you would see this. Although the length doesn't change, in an hour you're going to see the filament somewhere else, and it's spending energy for this. This is why it is called treadmilling. You know, you run on the treadmill, you spend lots of energy, and at the end you are going nowhere. It doesn't make a difference except you're burning calories. Okay. So just like there is the GTP cap in the case of microtubule, there is a similar cap here in the case of actin. Okay. The tubulin monomers, the monomers of microtubules, they are connected to GTP. The monomers of actin, they are connected to ATP. So if the hydrolysis of the ATP is slower than the polymerization, it's going to polymerize faster. If not, it is going to depolymerize from both ends. Okay. So there is no protective ATP cap here. There is rather the protective proteins, and those proteins can allow the polymerization and depolymerization of the actin filament. Okay, of course, again, in nature and in synthetic biology, there are so many things that are targeting the actin cytoskeleton. Phalloidin is a very nice stain. Here in the red, you can see phalloidin. It binds and stabilizes the actin filaments. So if you add a fluorescent tag, you can use it to stain the actin filaments. But in a cell, because you're going to see actins everywhere, sorry, this is the cell and actin is simply everywhere. Phalloidin is going to bind on the sides of the actin. It is going to stabilize them. So the actin cytoskeleton cannot depolymerize anymore. If it cannot depolymerize, it cannot change shape. If it cannot change shape, it is going to get harder. So this hardening is not good. It's actually how it kills the enemies. Phalloidin is extracted from mushroom, and whatever animal is eating this mushroom is poisoned and dies because of this actin actin stability. So it should not be more stable than it should be. And cytoskeleton, it caps the filament plus end and prevent depolymerization here. So it attaches itself here. So it cannot depolymerize. If it cannot depolymerize again, the treadmilling behavior will be disrupted. Because the treadmilling behavior is disrupted, it will not be able to react to the coming signals. And finally, there is latrunculin. It binds to the monomers and prevent polymerization. In this case, you're going to see lots and lots of monomers in the cell. They will not be able to polymerize. So all these actin monomers, they are useless if you cannot use them for a functional structure, a 2D or 3D structure. And this is what it is doing. So I'm sure you're going to find very nice, interesting examples. Okay, there are drugs, toxins, etc., but there are also natural actin binding proteins, ABPs, that are very useful in the cell. This is how the cell puts the actin together to make a shape. For example, thymosin and profilin binds and sequesters the actin monomers, so they will not polymerize. For example, this can be, or let's say, let's use another color. This can be thymosin, or profilin. When you have these proteins, the monomers are not used into polymers. So the monomeric polymeric ATP balance will shift to the monomeric one. Maybe the cell is going to create philopodia or lamellopodia soon. Remember these structures. It needs them in lamellopodia and philopodia, so it needs to increase the store of monomeric actin. Maybe it's using it for them, or maybe it doesn't want to spend energy. There can be many reasons for them. And there is formin and actin related proteins. They create actin, actin nucleation, let's say. There is an ARP23 protein. This was the protein I was working with in my PhD. And from this ARP23 protein, the actin polymerizes. It looks like actin, so it creates this polymerization. So wherever you activate this ARP23 protein, it is going to start polymerization. It is going to make the filaments. So most possibly here there are ARP23 proteins. I said that actin doesn't have a center like microtubule. In the case of microtubule, they all originate from the centrosome. There is no one single centrosome, but there is like hundreds of these ARP23 proteins which initiates polymerization. And then there are actin bundling proteins. Of course, how you bundle the actin totally changes the final structure you get. For example, in the philopodia, the actins are bundled side by side. So at the end, you're going to get a long and thin structure, which is this. So here they have this actin binding proteins, which attaches them side by side. Or there are some cross-linking proteins. They don't make parallel ones. They make this total random ones. And at the end, what you obtain is a 3D gel. Okay? And some proteins, by the way, cover the surface of the actin and they close the actin from interaction. Okay? So these side-binding proteins, they cover and hide actin. We will see them in the muscle structure. And finally, there are some motor proteins. The motor proteins can walk on actin, just like kinesin and dynein can walk on the microtubule. These motor proteins called myosin can walk on actin. Why? Actin is polar. Actin has a plus and minus direction. So whenever these motor proteins are walking, they can know which direction they are going. So here you can see some examples of filopodia and lamellopodia. Filopodia is like little fingers, tiny needle. Lamellopodia is more like a wave. And this is how the cell searches around. Remember, cells do not have eyes, ears. The only way they get information from the outer environment is to get the chemical signals and to touch. This is how they touch. Okay. Here you see examples of lamellopodia and filopodia, like little fingers and little hands searching around. And this is how the cell moves. The actin polymerization continues here. It makes an attachment. This is mostly a desmosome or hemidesmosome. And then rest of the cell is just dragged. Of course, after actin initiates this growth, microtubule accompanies it later. And here you can see some actin polymerization beneath this plasma membrane. There is lots of actin nucleation and branching, which makes this 3D gel. And there are some sphering proteins. The actin should be cut here into monomers, so these monomers can be added here. So depolymerization takes place here and polymerization takes place here. And some of them are kept, so it makes sure that they will not be depolymerized anymore. Okay, like this. There are cap proteins which ensures that there will be no depolymerization, so they will move and create a cap and a long thin filopodia. Okay, these different structures, they are controlled by monomeric GTPases. Now you know what a monomeric GTPase is. We have seen them in the chapter 16. How do you say? Molecular switches. Oops, sorry. These are raw class of proteins, raw family of proteins. Okay. This is called raw family, and there are three members: raw, RAC, and CDC2. They are all actin binding proteins. They all activate actin polymerization, and they all make structures made of actin, but these structures are different. When raw is activated, it makes contractile bundles. And you can really see this, these are really like muscles. You can see under the microscope that it is contracting. So it's getting smaller and bigger, contracting and releasing, contracting and releasing. Okay. When RAC is activated, it causes lamellopodia. It causes the structure under the plasma membrane. And when CDC42 is active, it creates thousands of these little fingers, filopodia. So please keep in mind they are all made of actin.